Okay, so this is the RNAseq with Bioconductor page. So we got a touch base with Sang, who says that their subjunct worked fine in the end yesterday once they actually used the full file. So it seems that somewhere in between my computer and GitHub, the sampled version of the file which we used in the exercises has got corrupted. So I'm gonna fix that. So if anyone's following this at a later date, hopefully it should all be good by then. If you're on the YouTube. Okay. So everyone should be all set up and ready to go, as it's the same setup, and we're going to go straight into session 3. And so in this session, it says at the beginning, we're going to cover things about working with large data sets. So you have many, many groups, and starting to visualize different aspects of these with PCA and clustering. So this is where I think things get really cool and exciting. I keep saying this. I hope you find it as exciting as I do, or at least interesting. Okay, so this is where we are. And just to recap what we did yesterday, we learned how to do alignment and how to do counting. And we kind of have two different methodologies for that. One where you do the full alignment and you then count, and you could go from a FASTQ to a BAM file, and then you have counts. And we used R subread and subjunct to do that, and then summarized overlaps within R to do the counting. Or the kind of direct, quicker, but not allowing you to actually get the alignment methodology, which is salmon, right? And that gets you straight from the FASTQ to the counting. And so both of these methods are very good, and they work pretty well. But they obviously have different features and benefits and costs associated with them. Subsequently, we then talked about how to do differential gene expression analysis using DE-seq2, what the different steps are, and some of the caveats associated with it, and some of the important statistical parameters, such as how it does shrinkage, and what does shrinkage mean, the shrinking of variance, and then also multiple testing to adjust for the fact that you're looking at 20,000 genes. And so if you don't do multiple testing, a lot of your genes will be false positives. So now that we have that kind of differential result and we have counts, we can start really looking at the data and ask what's happening to the data in a whole scale, what's happening to the data, and try and infer some functional relevance from that. And so to do this, we're actually going to switch data sets. So we're going to be using a data set which we used in one of the exercises. And so this is from Bing Ren's lab. And it's full of adult mouse tissues, this data set. And we have 3 different tissues that we care about: liver, heart, and kidney. And so if you want to go through all the steps, such as getting the FASTQs, doing the mapping yourself, and generating BAMs, you can do that if you want, okay? This is where all the raw data lives. And you can download from here the FASTQ files. You can download from here, I think, also the BAM files. So you can get a lot of the data directly from here. But you don't need to do that if you don't want to. We've provided the full counts that we made ourselves. It's in a range summarized experiment, and it's in your data directory. Okay, and it's called gctissuefull.rdata. And so we'll be using that predominantly. But just in case you want to know where the data is from, or if you want to use it, you can always follow some of these links. Okay, so as you can tell, we're considering a much more complicated data set, right? We have 3 tissues, and this is very common. Most of the time, people aren't just doing A versus B. Sometimes they are, and that's also still can be complicated. And even if it's a pairwise comparison, it's what we can still consider to be to have high dimensionality. And that's because you have so many like genes you're comparing across. You have many genes and you have then many samples that you're comparing across. And so trying to simplify down what the main trends in your data set are can be quite challenging. And so there's multiple different methodologies used for this. There's things like heat maps that partner with clustering. And so you can see this, we have this heat map here of several genes belonging to a specific gene set. And they've been clustered as well. So you have the specific patterns on a, have been clustered together in a row-wise basis. And so similar patterns are near each other. And so this can really start to help you understand what's going on. And then we also have things like PCA. And so it is chopped off a little bit at the bottom, but you have PC1 at the bottom, PC2 here. And we have a case where we have these different groups and they're all been colored. And you can see that most of the variation, if it hadn't been chopped off, is actually on this PC1. Okay, so it suggests that most of the variation is between this group versus this group. So using these kind of data sets, you can answer lots of questions, like what are the major trends in the data? How reproducible are my replicates? Like you can see the replicates here, much more, they're colored the same color. They're much more tightly close to each other than they are to other groups. And so that's an important quality control when you have multiple groups. And so being able to look at this and look at these more complex designs for experimental setups is really important to be able to go through these steps, first for quality control and then for biological interpretation. And something I kind of talked about last time is what we're talking about today pretty much directly mirrors some of the stuff that we do in our own pipelines and what we do in like further analysis when we're working with people. So it's very much the same kind of code. And so if you do work with us, you can see how we generate these plots. You can have a good understanding of that. Or if you want to do it yourself, you know that it's a tried and trusted method, okay? Okay, so to start with, we need to load in the data set. And so if you, hopefully everyone remembers, what we did last time is, I can show you, you would have downloaded the full GitHub data set. And I downloaded it, or the GitHub folder. And I downloaded it to my desktop. It downloads as a folder called RNAseq master, and it's a zip. I unzipped it. And then when I open up my R, I do set working directory. And I want to set to be inside the R course within it. And so if I use dir, I can see what's inside that. And the most important thing here is the data folder, the exercises, and the presentations. Okay, everything else is just, this is because I've been working with this project a little bit. Some of this we created yesterday and things like that. But yeah, so that is where you should be oriented. And so if you've got your RStudio set up like that, running this code should work fine, no problem. You'll just load it and you'll get the data. And when you load it in, you can just type in the name of the object. So this loads in an object, and that object is called gene counts. And you can start to infer things about it. So we have, in this case, we have 14,000 genes that we've tested or have counts for. There's 6 columns. And that's because we have one replicate or 2 replicates in each tissue. Okay, so 3 tissues, 2 replicates in each, 6 total. We have cold data information, which is our grouping. So this is our tissue information. And then we have all the gene names or IDs. And so with this setup, you can just jump straight into DE-seq if you want. We just say we have a DE-seq data set. We just want to do a comparison over the tissue. And then you can run the actual DE-seq operation. once you've generated the data set. And so this is just kind of doing a similar thing to what we did yesterday. And these steps work even if you have this much more, like this extra level of complication in your data set. It's a little bit more complex. We have 3 groups, but you can just perform the same steps. The change is now, if you want to get this kind of pairwise comparison, you want to understand what's the difference between liver and kidney, and you have a data set that has liver, kidney, and heart. You just have to specify which comparisons you want to do. And so in this case, we do the results function. We say it's on the DDS object, and we're comparing in tissue, and we're comparing liver versus kidney. And we can do this 3 times, and we can get every pairwise combination. Okay, and so this is the most simplistic way of starting to break down what's happening when you have more complex group information. If you have 3 or 4, you can just start to look at each pairwise comparison and start to get an idea of what's going on. And so in this case, we can look, we have heart versus kidney, and we get this result object, just the same as you would have done if you started with just 2 at the beginning of your DESeq object. Okay, and we have lots of significant genes, so that's good. This is promising. And so a common thing to do is to say, well, like I care about heart, for example. I want to see what's different, heart versus liver and heart versus kidney. Is there anything consistent? Is there anything unique to those? And so to be able to kind of ask those questions, it's quite simple data manipulation and data frame handling. And to do that, all we would do is convert these results objects. If we remember, this is a special result object. Okay, it's a DESeq object, and we convert it to a regular data frame that's very R friendly, using as.dataframe. We'd then do the filtering to remove NA values. So we showed you this yesterday. If you remember, when you do an adjusted p-value, there's often quite a lot of NAs. And what those NAs signify is generally genes that have pretty low expression. So DESeq doesn't even bother including it for multiple testing to reduce your total number to give you more power. And so you want to filter those out of the final result because they're not interesting, they're not relevant to what you're using going forward. Okay, and so then we have 2 data frames, and there's lots of information in these data frames. So in this case, we're actually subsetting it down and saying we're actually only want to look at the log2 fold change and the adjusted p-value. Okay, and so we're indexing here. And so for those who are feeling a bit rusty with the indexing for R, the indexing is a square bracket, and you have a comma in the middle. And anything indexed before the comma indicates I want rows. So you can put in like a number 1 and it will give you the first row. Anything in the second means columns. And you can use numbers for this, but you can also use, if the columns have names, you can use the column names. And so in this case, we're just subsetting out the column names. Okay. And so then I want to take these 2 data frames and merge them together. Okay, and so when we merge these together, we're able to then be able to have everything next to each other. We'll have heart versus liver and heart versus kidney, and then we can start to look at which ones have significance in both cases, which ones are significantly upregulated in both cases, things like that. We can ask those questions. And so to do that, we can just use the simple merge operation. But in this case, both of these have the same column names. So this is just something so you know, if you ever do merging and things have the same column names, it will get upset and it might rename them, but it will probably give them like bad names, like names that don't make sense. So what you can do is you can say, I want to get the column names from heart versus kidney, and I want to paste onto them heart versus kidney and an underscore. And so that means the column names will be relevant to what comparison it's from. And then I'll reassign that back into the column names. So we're just modifying the column names so they're actually directly relevant to the data frame they're in. And then we can do the merge step. Okay. And we're merging using the row names because the row names can correspond to the gene ID. And so that's why we can use by equals 0. And by equals 0 means I want to merge using the row names. I don't want to use a specific column. I don't want to use column 1 or column 2. I want to use the row names in both cases. And the product of this is we end up with a data frame where we have the row names. We're just looking at the first 2 rows here. We have the row names. We then have the heart versus liver log 2 fold change and its adjusted p-value. Then we have the heart versus kidney log 2 fold change and its p-adjusted value. And so, and this has been, it's kind of complicated. There's quite a few steps, but it is like basic data frame manipulations to get from this result of these many result objects to a kind of merged result object that contains multiple results in it. And having this makes it much easier to then make deductions about what genes are doing what in both cases. And so we can then ask the question, which of my table is upregulated log 2 fold change? So that means greater than 0. So we say, we use this logical operation. Which of these is greater than 0? And the heart versus kidney log 2 fold change is greater than 0. And the p-adjusted for heart versus liver is less than 0.05. And the heart versus kidney is less than 0.05. So with that series of logical operations, we're asking essentially what is significantly upregulated in both tissues versus the heart. And so with that, we get a index of true and false. And then we can subset our table. And then if we look at the first 2 rows, you can see how that's changed between this and this. Now, in this, most of the genes don't look that interesting. None of them are significant. It's like whatever. And now we have significance in both cases. And so we know that these genes are genes we care about for our tissue by tissue comparison. Okay. And so we now have, like, this is the kind of thing that you can then start to maybe export to a spreadsheet. You can share with people, like your PI, and then you can start to look through these candidates and like see what's going on, what's interesting and special with these, or you can follow them up with some other kind of analysis. Like in the next section, we'll talk later today, we'll talk about how to do things where you look for gene sets. And so you can see if anything's functionally enriched within this group. We do have a question from Charles. So can we use gene names instead of row names? Yeah. So I think I understand what you mean by this. So when we come to here, all we're doing here is merging by whatever's common. It doesn't matter whether you have your data frame labeled with gene names or whether they're labeled with gene IDs. It's just you want to find whatever's common between them to be able to stitch them together. As a convention, we use we in our group use gene IDs because they're a bit more robust. They're less subject to like issues with capitalization or not capitalized, or whether there's things like hyphens and things like that, especially when you look at multiple different species. Sometimes there's a much more complex nomenclature involved. for symbols. And so we stick with IDs because they're straightforward and much more consistent. And I generally recommend that for your bioinformatics. Use the IDs and then just add an extra column that has the symbols. But whenever you want to do any kind of operation like matching or anything like that, using the IDs is much normally a stronger way to go. I hope that answers your question, Charles. Yeah. Yeah. So for the merge, you can use whatever you want. So the key thing here we have to remember is these two data frames have 3 pieces of information in them. They have log2 fold change, they have the adjusted p-value, and they have a gene ID. And the gene ID is associated with the row names, and it isn't its own column. And so to do the merge, the only thing that's the same between these two data frames is the gene ID, which is in the row names. But depending on your data frames, so bi equals 0. So Charles followed this up. So he's interested in how this bi equals works. And if he's asking if it's in column 2, if the G name is in column 2, then you could use maybe bi equals 1. So it isn't a 0-based index, okay? And so what I mean by this is it doesn't start at 0, okay? So with these data frames, which is maybe tough it's gonna look a little bit like this. So column 1 in this case is base mean, but it does have row names associated with it. So these are like labels. They don't actually count as columns. And so if this was a symbol, for example, like a gene symbol, and I wanted to merge using that, I would use bi equals 1. But because I'm using row names, I do bi equals 0 because it isn't a column, it's a row name. Yeah, so in this case, I'm using bi equals 0. It then eventually, when you do this and you use row names, it gets converted into a column. So I can see how that output is a little confusing. And so at this point, once it's been merged into the full table, it is now it's its own column. But before it was specifically a label. Does that make sense? Cool, that's great. Yeah, and so once we have this table, and so yeah, what we've done here, if we remember, we've subset to specifically the genes that are upregulated significantly in both cases, and we have this data frame that contains all that information. We can then ask, we can make a logical data frame based on that to then put into a Venn diagram. So quite often when you do these kind of overlaps, you want to be able to make a Venn diagram to be able to understand what is the overlap between the two, what are the separate ones. And to do that Venn diagram, you often want a logical matrix, okay, that looks something like this. So this is going to be the output, where it's false, true. And so in this question, we're basically asking the same as this. So this is how you create the subset. But in this case for the, yeah, so this is, let me re-summarize. This is what I do if I want to say export this into a spreadsheet or something like that. I'd create the subset and I'd export the subset. Or if I just want to work with it directly. If I want to understand what's happening with both by using like a, what's it called? By creating a Venn diagram, I would still use the full table. And I would just ask the question, which one in this case. And so which one in the up versus liver is greater than 0 and has significance? So full table, P adjusted, less than 0.05. Okay, and so that will create column 1. And then I'll do exactly the same again for the kidney comparison. And so starting at this full table, we're able to ascertain like which ones are significant in up versus liver and which ones are significant in up versus kidney. And then we can use the lemma package, which has a Venn diagram function. And so if you have your result formatted like this in this logical matrix, you can then just supply it straight to the Venn diagram function. And so you can see in our case, we have 2,000, just over 2,000 genes, which are upregulated in both cases. But you have just like almost 1,500 in liver and 1,100 in kidney. And then you have a group of 9,000, which just didn't pass criteria in either case. So I'd say this is like the most simplistic way of starting to understand these complex data sets, is doing things like these overlap analyses. We get often asked to do them, but I would say they are the most kind of the lowest hanging fruit. And there are some caveats associated with this. And so if there's some specific, sometimes what we see is maybe there's some effect that one group has some specific differences in variation. That makes doing this kind of analysis a lot more tricky. And so what I mean by this is, say your liver sample had a ton of variation within it. It becomes a lot harder to then draw conclusions or differential gene expression analysis between that sample, so the liver with the high variation. It's going to look like there's a lot less significant differences between, say, heart and liver versus kidney and liver. And so if you're doing like this three-way analysis, it might look that the differences between heart and kidney are like there's much more differences between heart and kidney compared to the comparisons with liver. It might seem that liver is much more similar. And that might just be because the underlying data is much more variable, and that could be a technical thing. And so with these kind of low level analysis, with these kind of overlaps and Venn diagrams, things like cutoffs become much more important. And things like the quality of your data can actually infiltrate in and affect the conclusions. So it's something you have to think about and understand before you like draw these conclusions. It's important to understand your data in a lot of detail. And that's why we kind of show you how to look at things like the variance for these pairwise comparisons and things like that. Oh, yeah. Anindita had a question, does up versus liver mean genes that are upregulated in liver compared to heart? So we can just double check that. So the comparisons that we used for this were the heart versus liver, or heart versus kidney. Okay, and so in this case, the comparison is considering the heart to be not the baseline. So the baseline is liver or kidney because it's the first one. Okay, so it's the numerator in the log2 fold change. So if you see something that is a log2 fold change greater than 0, that means it's a propensity for being in the heart. And then when we have a look here, everything is greater than 0 that we filtered for. So yeah, that means we're looking for genes that are more enriched in the heart than either liver or kidney. And so, yeah, we have, this would be what I would say, like, this would be like your cardiac signature genes if you were like to compare between these two. from this specific example. Okay, so that's kind of like some low-level analysis. And next step would be to start to visualize your RNA-seq data a little bit in a more nuanced way. And one of the first things we want to do is actually look at counts. And quite often you want to look at counts for a single gene, or you want to look at counts for like a group of genes. But it's quite often actually tough to visualize this in a sensible way, because it's an integer, and the variation between your genes is quite large. And so you'll have quite like some genes which have 10 counts and other genes which will have 1,000. And so being able to compare what's going on between all these different situations, you often put things on a log scale, because it then becomes much easier to visualize what's happening. And so to be able to do that, there's some functions built into DESeq to help you with this visualization. One of them is norm transform. And what that will do is do a log2 transformation, but it will add 1. And if we remember, whenever we're logging something, we'll probably add something beforehand to deal with 0 values, so it doesn't error. And once we do that, we can use the same assay function to extract out the counts. Okay, so this step just did a transformation. You can see it's now a DESeq transform object. We can do assay to get the counts out, and then we can provide that straight into box plot. Okay, and so we have the transformed counts, and we have the it's shown in the box plot, which is, it's cool, but pretty much it's very consistent between all of them. It's generally what you expect to see if you're doing a genome-wide visualization. You'd expect everything to be fairly consistent across the board. And so something you face when you start to want to like put counts out, start to look at counts, is that you face a similar problem to when you do the differential analysis. And that's because the smaller counts have a higher variance. Okay, and so we've talked about this low counts, high variance as a problem. And so this may actually mean that your smaller counts have more influence over just how you visualize things, or if you do subsequent clustering, they can have a stronger effect. And so you can see here, this is the standard deviation versus the mean expression, but ranked. Okay, so strongest expressed up here, lowest expressed down here. And so you can see there's much more variation here. And so one way to deal with this is to do an RLog transformation. So this is essentially doing a log2, just like we just did, but it also does some of this variance stabilization of these low-end genes. And so it makes it a much more similar count value to the one that's actually going into your DESeq analysis. And so that's why we like using it for a lot of different approaches, because if you're going to be talking about DESeq, but then you also want to do some heat maps of the counts, using the RLog means everything it's basically using a very similar set of inputs. Okay. And so to do this, this is actually this can, depending on your data set, take, it's one of the more longer times to wait for it to process, but it never takes more than a minute, really, unless you have a massive, massive data set. And so we just do RLog. And again, we export this to a new variable called RLog tissue. And what you create is it's a DESeq transform object. So it still knows it's DESeq, and it's known it's undergone a transformation. And most importantly, I think why we use these objects is it still retains all the information, right? It still retains all the DESeq information here about dispersion and stuff like that. And it still has like tissue and column information. Okay, we have a question from Jesse. So we are only using VSN here, so I can do this mean SD plot. We don't really use it for anything else. So if it's not installing for some reason, We can have a look at that later to see if we can figure it out. But it might just be, it's not a super big deal. This is just as a demo of like what the data distribution looks like. It's a nice plot that's easy to do. Okay, so once we've done the RLog, if we compare we do assay to get out the counts, and then we can put it into the same SD plot. And we can compare that to our matrix of norm. And what I hope you can see is the axis, broadly the graph looks very similar. So we haven't really changed the data too much. We haven't transformed it or done anything. We have transformed it, but we haven't done anything to really warp the data in any way. But you can see the axis has changed quite dramatically. So we've cut about 1/3 of the variance from the low end in this case. And really the variation has also dropped throughout. But it has the biggest difference at the low end. Because we're going from about 0.5 here to 0.4 here, but we're going from 1.6 here to maybe 1.1. Okay, so it has a more dramatic effect. Yeah. Are RLog and LFC shrink similar? So conceptually, maybe a little bit, but actually they're quite different. Okay, so maybe that's not a good answer. So if we think about what RLog does, RLog it's transforming your counts. Okay, so it's the important thing that with RLog, it's all about counts, and it's transforming them on a log2 level, and it's shrinking the variance associated with predominantly low-level counts. Okay, LFC shrinkage is shrinking the log2 fold change values of genes that do not have really much significance associated with them. And so what this does is it downweights the fold change of genes that have very little significance in their in the comparison. And so what that does is it allows you to then use this kind of log2, the shrunken log2 fold change values as like a kind of interim methods where it considers both the amplitude of change and the significance of change. And so that can be really useful for things like GSEA. Yeah, and so the big thing is one is dealing with this standard deviation or the variance, and one is dealing with significance, right? And so again, we have the kind of interplay between counts and variance with RLog, and we have the interplay between log2 fold change and significance with the LFC shrinkage. Okay, both of them help strengthen your data then. And they both have very kind of, I'd say, specific use cases. And so LFC shrinkage is really good for setting you up for GSEA. RLog is going to be really good for helping you get data ready to do data visualization of counts. And so we'll show you what that goes into. And so the first thing I often do, especially with a data set like this, but pretty much any data set where you have replicates, is I want to do PCA. Okay, and so this is dimension reduction. And so hopefully most people have a rough idea of what PCA does or has seen a PCA plot. But really the idea is, like normally in a data set, you have thousands of genes with tons of different samples and groups. And really that makes it hard to interpret what's going on a global level. You want to distill the general trends or distill down to general trends in behavior. So that trend could be something like a lot of the genes are being upregulated in heart, for example. And so you try and find the patterns that are consistent across your data set. And so that these are considered like meta or eigengenes, which kind of represent major patterns across your samples. And so you use PCA to find what are the strongest patterns. There are many different forms of that. There's, PCA is the one we'll talk about, but there's also multifactorial scaling, non-negative matrix factorization. There's also some which are used very commonly in like single cell. So you've probably heard of T-SNEs and UMAPs. And so there's lots of different kinds of doing it, and each of them have their own use cases. But for your general bulk RNA-seq, PCA is the go-to. And so this is an example, right? You have this whole group of genes that are all doing this similar thing. And there might be also hundreds of thousands, not hundreds of thousands, there might be another 5,000 genes doing something else as well and doing a different pattern. But we want to distill out just the key patterns into a single trend that we can observe. And so this is what this looks like with our heart and kidney and liver. When you have created your RLog object, okay, so this is the RLog object that we created. So this is, we just took the DDS object and then we did RLog, and then we can just do plot PCA and it will tell us, show us the results. Okay, so it's super simple. It's built into DESeq2. And all we need to give it is we can give it the metadata group that we care about, and then it will separate the colors and create a nice key for us straight away. And you can also, if you want, specify how many genes it considers. So at the moment, I'm using nrow for the entire RLog. So this is a PCA of the entire data set. But by default, it only uses the top 500 genes. And what I mean by the top 500 genes is the genes that are contributing most to the patterns observed in the data. So if you have a bunch of genes that just don't do anything, they're consistent across the board, they don't do anything interesting, they'll be low down on the list. Okay, and so what we have here is 51% of the variation is in PC1. And so what's happening on PC1 is really this separation of heart and liver, and kidney is an intermediate that's in between the two, right? And so over half the variation is in that kind of axis. And then in PC2, it's the separation of kidney versus the two other tissues, okay? So genes associated with PC2 will actually be very similar in heart and liver, but they'll be very different in kidney. And so between these two PCs, this accounts for 95% of the variation. So this is actually like the majority of the variation that we would care about in this data set is actually one axis here and one axis here. You can go into deeper PCs like PC3, PC4. Sometimes we dig deep because to do this, to try and understand other parts of the data if you have a very complex data set. But in this case, it's pretty simple and most of the variation is already accounted for. Yeah, and so it is potential that there are genes in PC. So it's not. I think we'll dig further deep into this to kind of help answer the question. But the genes driving that are predominantly responsible for PC1 are not super likely to be the ones also driving PC2. However, every gene contributes to a certain amount, either positively or negatively, to a specific PC. But it might be 0.000001. Okay. Yeah, so this is kind of what I go into. Most of the variation is covered between PC1 and PC2, and they have inherently different things that they're contributing to. And so this function that we used, this plot PCA, is a really nice way just to visualize what's going on. But quite often we want to dig further into this. And that plot PCA doesn't actually give you the ability to dig into it. And so what we do in that case is we use the PR comp function. And so you actually have to use T to transpose your matrix, because it wants, when you do PR comp, it wants all the things that you're considering over. So in this case, the genes, it wants those to be on the kind of X or the columns. And so what the transpose does is it rotates so that the columns are rows and the rows are columns. And then you give this to PR comp. And what this does is the PR comp function is basically doing principal components analysis, but it gives you access to all the kind of underlying workings. And so you can start to interrogate what's happening at PC1 and what's happening at PC2. Okay. And so we can do PR comp. We can have a look at the X values. And these are what, these are the mappings, okay? And so what we mean by mappings is, you can see here we have PC1, PC2. These are the coordinates that we use in this plot, okay? And so if you look at X, you can try and generate your own version of this plot. And we're only looking at the first 2 rows here, but it will have a row for every single sample. And it has all these other PCs as well. So if you wanted to look deeper at PC3 or PC4, you could look at them and graph them yourself. And we can then plot this ourselves, and we're basically recreating what we saw before with the plot PCA function. But now you have the ability to customize it yourself. So if you didn't like the colors, you didn't like the scales, things like that, you can generate it yourself using the plot function. We provide PCRes as the data object. Okay, so we're giving it the loadings. And then we're saying the specific columns that we want to care about. Okay. And then we create a legend. Okay, so this is some just plotting specifics. If you want to get into the minutiae of plotting, you can. But essentially what we're doing is just recreating the plot that we've already seen. Is there a way to put back the variance into the axis? Yes. So you can interact with PR comp and extract out the variance from PR comp. I'm not sure if we covered that here or not, though. If we don't, I can dig that out, or you can just Google how to grab it from PR comp. But it's contained. PR comp has lots of things beyond just the mappings. Yeah, one of the things it does have, so this isn't relating back to the variance, but this is why the predominant reason why we might do PR comp ourselves is to get the loadings, okay? And so the loadings or the rotation is how much does an individual gene contribute to a PC? And so the genes that have the highest absolute value in the PC are going to be the genes really driving the differences you observe in the PC. Does PR comp always return 6 PCs? I'm not actually sure. Most data sets, if you're going deeper than 6, then most of the time you won't really be looking at anything that's that interesting after 6. Most of the significant parts of the data would have been covered already. However, I wouldn't be surprised if you can ask for more. And I know this isn't the only way you can do PCA analysis. And I know with other packages, you can go down to 20 if you want. You just specify. But I haven't specifically, off the top of my head, I can't remember doing that with PR comp. Okay. And so what the most negative values on here would, in PC1, would be the genes that are most heart-like, okay, on this specific PC. And the most positive genes in PC1 would also would correspond to the ones that are most liver-like, okay? So using these kind of loadings, you can actually start to get similar kind of, it's an alternative approach to be able to finding the genes that are really driving expressions or driving characteristics within your data set. And so sometimes we experience cases where actually it's very hard to, the differential gene expression isn't working very well. Maybe there's quite a lot of variance, but despite the variance, we can actually see a PC that actually explains what we're seeing. But there's a lot of variance in one of the other PCs, which makes it really hard to do the differential gene expression. Maybe there's a batch effect or something like that. We can use the PCs in those cases to start to identify, like, what are the candidate genes? What are the genes that are interesting when the differential gene expression isn't working? So this is a tool we lean on for things like that. Yeah. And so in this case, I'm showing you an example where we want to investigate the kidney samples. So if we remember, PC2 is really driving kidney versus everything else, okay? And kidney is specifically negative. So here we're going to grab from PC2, because we're getting the second column from the rotation, and we're saying decreasing equals false. So that means we actually want increasing. So it's going to, we're sorting by negative, like we want the lowest values first, and we're going to grab the top 100. Okay, and so we can get this gene ID, and I could actually, let's do Monterey. Oh, my internet's been real slow. Yep. So if I went to gene, I search for 20505. Solute carrier, family 34. That sounds like something important for kidneys, right? So using this can be super useful for like identifying these differences. And then you can start to do some things like visualize what's going on. So in this case, we have these PC2 markers, right? So these are our top 100. And this is a little circular, what I'm going to show you, but it makes sense. So we can look at our heart versus liver, our heart versus kidney, and our liver versus kidney comparisons. And we're basically extracting out or subsetting specifically to those that we think are driving the PC analysis. Okay, and we're doing it for the stat value. So this is kind of the directional statistics out of the log2 fold change analysis. And you can see that really, you can see that they're all being downregulated in the comparisons to kidney. So that means they're more prevalent in the kidney because of the way we're doing the comparisons. Okay, so it is a little bit circular because we asked the argument, like we looked at the PCA, we had a look to see which ones were the most driving the PCs, and then we fed that back into DE-seq2 results and looked at what ones are driving the DE-seq2 results. But they're two different statistical approaches, right? And we're subsetting our results with the PCA, and then we're looking at the results from the DE-seq, and you can see that there is consistency there, right? There is no real change on the heart and liver level, which matches what we saw at the PCA, and there's a much more drastic change in the heart versus kidney and liver versus kidney, and it's quite similar between the two, which again matches the PCA. Okay, so unsurprisingly, I guess, I thought I was going to be on time today, but I'm running quite far behind again. So sorry. We'll soldier on, and this next section is pretty straightforward. And then there's a little bit about clustering. So we're just going to kind of start to touch on clustering. And I think it, again, like I said, I think it's fairly straightforward. So it shouldn't take too long. Okay, so another important quality control, which is very similar to PCA, is to do a sample-to-sample correlation analysis. And so you want to know, do my replicates, are they more similar to each other than anything else? And when you have multiple samples, sometimes you want to, or multiple groups, you want to ask the question, is group A more similar to group B than group C? And so to be able to do this, we can do some correlation on our RLog matrix. So again, we're using our RLog matrix, and we just do the core function. And what this would then do is it gives you an idea of the correlation between each of the samples. Okay, and so sorted heart 1 versus sorted heart 1 has a 1-to-1 correlation because it's against itself. But you can see that sorted heart 1 versus sorted heart 2 has a 0.98. So that's actually also super high, which is good because this is the same tissue. It's just a replica. And so once you do this, you'll then want to be able to plot this into a heat map. And so what we often do is convert these into distance scores. Some people just plot correlations. Other people prefer a distance matrix, okay? And so to do a distance matrix, you do 1 minus the correlation, and then you could do as.dist to convert it to a distance object. Okay, and so correlation and distance are very, very similar, but they actually have like almost opposite values, right? Because if something is very correlated with itself, it has a score of 1, while if something is very distant, it has a score of 1. And those are kind of opposite meanings, right? Because if it's very distant, it means it's not correlated. So that's why we have to do the 1 minus. We can then turn it into a matrix doing as.matrix. And so we now have a sample distance matrix. And when you have that, you can then just put it into pHeatmap, okay? And the nice thing about doing it with pHeatmap, the clustering can then be controlled by the sample distance metrics that you provided or you calculated, okay? So if you remember, we have this sample dists here, and then we created a matrix from it. So we can use these distant metrics, and we'll use the matrix itself. So when you do pHeatmap, you give it the matrix, and so this is what it's going to use to plot the heat values. And then for the clustering, so that's the hierarchical trees on the sides, you can give it the sample dists. Okay, and so you can now see that the really blue has very little distance from each other. And so that means they're very similar. We don't really like that matrix though, because it's kind of counterintuitive to color scheme. The default color scheme in pHeatmap it works fine, but actually most of the time I end up changing the color because it isn't quite what I want. And so one of the ways we do that is with our color brewer and brewer POW. And in this case, I'm just saying I want blues. And I want 9 different blues. And then actually I'm going to create a gradient. So I'm going to do a ramp palette. And I want these 9 blues to be divided into 255 different colors. So this gradient is actually just 255 different colors going from this very white to this dark blue. Okay, and so this is what this gradient looks like. I can do plot to have a look at it. And so we've saved this gradient as colors, and we can just supply that into this. And so we can put the color equals colors. So that's an argument within our p-heatmap. And so everything else is exactly the same. And now this looks really nice and pretty. Okay, so we can see that there's a lot more similarity here. And actually, although it's putting kidney and liver very close to each other, or closer to each other than heart, really that you can see by the height of the trees that everything is very dissimilar to everything else. You can also add column data information to p-heatmap, which is nice. And so that goes into a variable called annotation col. Okay, and you can basically extract out the col data information directly from your DDS object. You turn it to a data frame using as.dataframe, and then you can supply that directly. Okay, and so now you can see it's labeled with the tissue. And also contained with this col data information is also the size factor. So this is part of the normalization step. And so this can also be interesting because it could actually correlate to something. You can get rid of this if you want for your final result, but when you're doing quality control, sometimes that size factors can be relevant. Okay. So that was the quality control. We, by default, for every pipeline run, we do PCA and we do this sample to sample correlation matrix or distance matrix. And that's what we do with every single pipeline run. The next step when you want to investigate your data is clustering. And we don't do this by default because there's a little bit more nuance associated with it. Okay, but I think it's a lot of the time worthwhile doing, especially if you have a pairwise analysis, it's less interesting. But if you have an analysis where you have at least 3 or more groups, it's really, really, really useful, especially time courses. If you have a time course, it can really be very illuminating. Okay, so to get into this, we can use pretty much the same methodology we just talked about. And that is p-heatmap. So p-heatmap can do the clustering for you. In the last case, we provided the distance metrics we wanted associated with the clustering, but you can let it take care of it for you if you want. But a big kind of conceptual step before we can put something into p-heatmap is for clustering to work, you often want to remove as many genes as possible, okay? And the reason being is if you do clustering and you're looking at a bunch of counts, so what goes into your clustering is count data. If you have a bunch of counts, like we have the RLog counts, if you include all the genes that aren't changing, the majority of your clusters will be this kind of like genes that you don't care about. And so what we can do is we can perform a test similar to an ANOVA, called an LRT test, a likelihood ratio test. And what the LRT test does is it allows us to say, is there any genes that have very low intra-group variation, but quite a lot of inter-group variation? Okay, and it does it over all of your groups. And so it's not giving you this kind of pairwise log2 fold change, but it's saying, these genes are significantly changing at some point in your data set. And so it does it by, you have this 2 different models. You have this idea of a tissue model. So that's your groups, essentially. In this case, we have our liver, heart and tissue, heart and kidney. And then we have an idea of a no groups model. Like that's when the no groups exist. And so you're asking, like, is there a functional, like if we look at our data set, is there a functional difference between these 2 models? And are any genes kind of breaking, like leaning towards this kind of groups model? And so to produce a LRT test, we just go back to doing DEseq, okay? And we can just take our original DDS object, we change the test. By default, it's doing up this pairwise test. We change it to doing an LRT, okay? And we're saying we're doing a reduced model with the tilde 1. And now because this is performing a very specific kind of test, we don't want to just assign this back to a DDS object. So we create a new DDS. We're very inspired and call it DDS2. And because there's no kind of, again, pairwise comparisons or anything, we don't need to specify anything in the results. So we can just call the results function and have a look at that. And again, we save this as a results object called across groups. We did something similar yesterday where we order it by a p-value so that we can have the most significant ones at the top. And if I look at the first 3 rows, you can see that we have some very significant genes. It gives you log2 fold change and things like that, but generally I just ignore this. It's all about the adjusted p-value. Okay. And so, as you can see there, the p-value is in this idea of comparing these 2 models, one where no groups exist and one where it's considering the tissues. And so if I was to take 1788, this is the second most significant gene. If I was to do plot counts, okay, this allows you to visualize a single count. You can understand why this one is maybe significantly different. And that's because it's really upregulated in the heart, okay? But by doing this analysis in a single test, we're just getting all of the significant genes. And it could be that it's really significantly up in the kidney. It could be really significantly up in the liver or downregulated in one of these tissues. And it's all coming from a single analysis because it's similar to an ANOVA. And so now we know all of the genes that are significantly changing. We can subset our data set to just those genes. Okay, and then we'll use those for the clustering. And so we subset in a very similar way to what we've subset before. We ask which genes have a significance. In this case, we're going to do a 0.01 cutoff. We ask which genes are significantly less than 0.01, and then which ones also are not NA. So we don't want to get any NAs in this, and we extract their row names. And then we can go back to our RLog matrix, and we can ask the question, which of our row names are in the significant changing group, okay? And so we're then subsetting our RLog matrix to be this new significant matrix. And so it's still a matrix that contains transformed RLog values, but it only contains them for 8,000 genes instead of 14,000 genes. And these 8,000 genes are all significantly changing in some way across your data set. So we basically just got rid of all the crap we don't care about, and we just have a data set of like interesting genes at this point. And so we can then just give it to pHeatmap. And so we often do this. We'll give this matrix to pHeatmap, and you can see everything is changing. One of the key things we do here is we do scale equals row. And what this means is for every gene, instead of just showing you the count values, it does a z-score analysis. Quite often when I talk with people who are a bit naive with bioinformatics, they're like, why can't I just plot the count values? I want to see what the counts look like in this heat map. But some of these counts are going to be thousands and thousands, and others are going to be hundreds or 10. But they might actually, across your liver, heart, and kidney, they might be behaving very similarly. So you might, in these three genes where you have 1,000, 110, they might all get upregulated in kidney. But unless you do something to normalize to the levels, you can't tell that they're actually behaving very similarly. And so by doing a z-score analysis on a per gene level, it allows you to just focus on trends and not absolute values. Okay, so this is nice. And you can also see it's drawn some trees. No, so yeah, Charles asked a question. So the heat map is not absolute value normalized per row. It isn't. It is normalized per row, but it's a z-score. So that is kind of how many standard deviations from the mean are you, essentially. And so, for example, in this top group here, there is in heart and liver, there is less counts in this top group, but more counts in the kidney. So these will probably correspond to this PC2. Yeah, do we expect these clusters to mirror PC groups? Probably. So if you're doing a really thorough analysis, quite often people, and you're looking at clusters and things like that, quite often people will compare different statistical methodologies and make sure that there's some like good rationale and they overlap. And so we kind of touched on that a little bit where we looked at the PCA and then we looked at the results of the DE-seq. But you could then also look at results of clustering and see if all three of them overlap. That would definitely be like super, super thorough methodology. Yeah. But yeah, I think, yeah, and so they're touching on the clustering. You can see here that we have trees both in the y-axis and the x-axis. And the y-axis is, the x-axis is, it's very similar to what we've seen before. The samples are grouping together. And then the y-axis, we have all of those like trees here. And this, so by default, it does hierarchical clustering to group these genes together into patterns, okay? And so you can start to break these down by just looking at them. And so if I just visualize these, I can make a guess at how many clusters look sensible, right? I could like say there's one here, maybe there's another one here, maybe another one here. And you can kind of start to get an idea of how many clusters and how many patterns seem sensible. Okay, and here's just a zoom in. And so quite often, like, as I said, Heatmap uses hierarchical clustering as its default. There is a ton of methodology included within the help for how to do hierarchical clustering with and extract out specific numbers of clusters with Heatmap. But we're actually going to focus on k-means clustering for this. They are both good clustering methods, and I use both regularly. I'm not going to say one is necessarily better than the other. They work in different ways though. So hierarchical clustering works on this idea of kind of you're dividing, right? So you start at the top and then you divide into two, then you divide into those two into two, and then you divide those two, those four into two, so then you have 8. You keep branching out. What k-means is you're sticking together, and you start off with a centroid and you say which is closest to my centroid, and then you add another one, and then you reassess where the centroid is, and then you add another one, and then you reassess where the centroid is, and you optimize out. Both can work very effectively. If you're not sure what to do for your data set, you can always talk to us, but I'd say most of the time it shouldn't matter too much, but you can run both to do a comparison. But you can still do the k-means also with Heatmap, okay? And so the nice thing about this is your statistical analysis is tightly tied to the visualization. And so it's a nice conversation between the visualization and the statistics. And so at the moment, we just kind of pick a number. So we pick 7 as a reasonable number of clusters, because they look like there are maybe about 7 patterns to major patterns within the Heatmap. And so I can do k-means k equals 7, and I'll assign this value out. Okay, and when you do this assignment out, it doesn't draw the heat map anymore necessarily, but I can, yeah, it depends on your settings within your R. Sometimes it will draw it out and sometimes it won't, but you can also press just enter in K when you assign it, and it will show you the heat map. And so what you can see here is the results. And so instead of seeing every single gene at this point, you see every single cluster and what that cluster is doing. And so we have cluster 3, which is up in heart and up in liver, but down in kidney. Well, you have cluster 1, which is up in heart and down in liver and kidney. Okay, and so you can see we have all these groups, and you can see how many genes belong to each group. Contained within this K, apart from that image, is actually the cluster information. So under K, there is a slot called K-means, which you can access with the $. And then under that, you can just keep going down into cluster. And so if you have a look at what's inside this, we can convert it to a data frame. And it basically has every single gene with the cluster associated with it. And so here we're just showing the first 10. And so, yeah, we have every gene and every cluster associated with it. So now you know the gene IDs, you know the clusters, and in a separate data frame, you have all the counts. And in another data frame, you have log2 fold changes. And in another data frame, you have PCA values. So you can start to stick these together and do comparative analyses this way. Something we like to do with this, once we've done the K-means, is actually go back and look at the gene values, right? And so when we looked at this heat map, it was summarized up. But I want to look at how it looks with every gene included. So to do that, you can take this matrix, and then you can order it using the clustering information from the cluster analysis. Okay, so you can order it using this. And then when you do this reordered matrix goes in, you can have a look at the matrix then, or the heat map. And so you can see it's now separated nicely, and you can see the patterns across and get kind of a more global vision of what's happening with this clustering. A key point when you do this is you want to turn cluster rows off. Otherwise, it's just going to be clustering the rows. Okay. So we're nearly there. So the last thing I'm going to tell you about is optimizing your clusters. And so when you do this kind of analysis, you are, we picked like the number 7, right? And I think it is a valid approach to look at your data, look at what it's telling you, look at the patterns and go, these look like an appropriate number for my experiment. And I'd say at least 50% of the time, that is the major way I decide to go. But sometimes it's harder to decide on a number. And there are statistical approaches to help you understand which is the most statistically optimized approach. And the way these work is there's a bunch of different methodologies for this. We're going to show you how to do a silhouette analysis. The way a silhouette analysis runs is it will look at the variation or the similarity within your cluster, and it will compare it to everything else. And it will try out every single clustering option. So it will try out what happens if I do 2 clusters, what happens if I do 3 clusters, what happens if I do 4 clusters. And it tries to find this kind of maximize the relationship so that you have the most intra-cluster similarity and the least inter-cluster similarity. Okay, so all the groups are all the clusters are very like each other, or like themselves, but not like each other. And so, yeah, the math is explained here. I explained it already. And so to do a silhouette analysis, there is a package called mbclust, which we use. And so what first we want to give to mbclust, when we do clustering, we want to give it the scaled values. And so this means the z-scores, okay? And so to be able to get the z-scores, we first have to transpose significance matrix. We then give it to scale because we transpose it so it can do it on a gene by gene basis, because it wants our significance matrix is rows with genes, but the scale works on a column basis. It's just a nuance with these tools. But eventually, when we do the clustering, we're going to want it to be back with the genes on in the rows. So we do a transpose, do the scaling, and then we transpose it back. So that's why there's these nested T's with the scaling in between. And so this scale function is creating the z-scores for us. Okay. We can then supply this to NB-clust. And there's lots of different options for this, but we'll just pick the distance for the optimization. Okay, would be Euclidean. The minimum number of clusters we want to test is 2. The maximum number of clusters we want to test is 12. We're doing k-means, and we're doing silhouette. Okay, and you run this. This can take a little while because it's doing clustering 11 times. But it does an assessment of how good each of those clustering is, and then it tells you which is the best. And so it says the number of clusters that's best for our data set is 3. So 3 is a great number. It's good, but depending on how deep in the patterns you want to understand on your data, 3 might not be enough. And so this is why I think it's always good to assess your data, but it isn't always necessary you have to live by what it says when it says 3 clusters, because you might be interested in some other deeper clusters. And so we can do something very similar to what we did before, which is we can take this clustering information, we can then apply it back to our heat maps, and then look at the ordering. And so the best partition section of the cluster num is contains all the clustering information. And then you can, similar to match, similar to merge, there's a function called match, which works on single vectors instead of matching data frames or merging data frames together. It matches. Ooh. Did someone have a question? Oh, no, that must have been an accident. Okay, and so if you want to match 2 vectors together, you can use match. And it will tell you which positions in vector A matches which positions in vector B. And so by doing this, we can then use the names of this to reorganize our original matrix. And then we can plot that. And so this original matrix, we kept the row names, we kept some annotation information based on the original clustering we did with 7, but we've organized it in a way that it's representing our new optimized clustering, which has 3 clusters. And so you can see there's a lot of overlap in how these work. And so like this one really contains cluster 4, and I think it's cluster 7 as well. Okay. Well, this one is cluster 3 and it's cluster 1. Okay, but it also is a single cluster from our optimized clustering. So by doing kind of like comparing some of these clusters, you can see how some of these clustering approaches maybe overlap and interact with each other. And when you I'd say this is quite common when you do optimize your clustering, you will find that you quite often get the number of groups that you have. And so looking at this result, right, we basically find that the number of clusters it found interesting as the optimized amount is It's the ones that are upregulated in liver, and the ones that are upregulated in heart, and the ones that are upregulated in kidney. And we know there's some more complicated other subgroups within that. And so maybe 7 might be a better number depending on the questions you want to ask. Okay. But maybe this optimized number is exactly what you need. So it varies. So I've really big time overran. I don't expect anyone to finish the exercises in this time. These are the wrong exercises. Okay, so this is linking to exercises 4, but we're in 3, so that link is wrong. But you can access them through here. Okay, but yeah, like I said, I don't expect everyone probably needs a break. I will come back though in 15 minutes and start talking about the exercises, just so that we have a recorded version for when you want to look back at this. Okay, I'm also going to stick around if anyone has questions in the next 15 minutes before I do that. That's totally cool. And if you are just loving what we're doing and want to dive into the exercises, be my guest. But again, there's no pressure. I hope that wasn't too much. It's the first time we've run it in this structure, and so it may have gone a little long because I haven't quite got it optimized yet. But I also get the there hasn't been much time to do the exercises. So if you want to just follow along, that's cool. If you're just having a break before we get started, I'm hoping to get all of this done by 3 so that we can talk about gene set analysis at 3. So yeah, so hopefully everyone can see my screen. And yeah, so what we run through here is we're basically in a lot of this can be running very, very similar, if not exactly the same as in the slides. I actually should modify this so we use a different data set, just so that there's a little variation. But for now, we're using exactly the same data set with liver, heart, and kidney from the Bing Ren lab. And the first question I ask is just load in the DESeq2 object for the tissues experiment, produce an RLog transformation, and plot the PCA. And so, as before, I've told you that the data exists in this in the directory. It's this object here. Okay. So we can do library, and the nice little wrinkle here that that's slightly different to what we did before is, I'll get to it at the end, is in the plotting. So if we can do load, we'll load in our data set. I'm going to run DESeq dataset and run the actual DESeq operation. And so you notice I loaded in the library for ggplot2. And the plot PCA function, for those of you familiar with ggplot, the DESeq plot PCA function is actually using ggplot to visualize this. So if I can do that, oh no, I just did that for a second time. Let's grab that. Okay, so yeah, for those of you familiar with ggplot, this is a ggplot. And so if you're not familiar with ggplot, it's probably the best graphing package there is for R. It's one of the main selling points for people to use R. It's part of the tidyverse. And so I would encourage, we have a course about using ggplot. I'd encourage you to check that out if you're unsure about how to make plots look nice in R. It's a really good set. But because it is this function uses ggplot, we can actually do things like add in ggplot themes. So I might not like this. I might not like this theme where there's these gray boxes in the background. So I can just do theme black and white, or I can do theme min minimal. Okay, and so there's lots of different themes. You can install themes and do all kinds of things depending how you want your plot to look like. You can also change things like the axis and stuff like that. And it all just needs ggplot style commands to be added on top of this plot PCA. So you can do a lot of customization without actually having to do your own version of like PR comp and things like that. Yeah, so that's applying the PCA. Next, we do the LRT test. So if we remember, we just do test equals LRT, and we do it reduced to tilde1. So we're creating this alternate model. And then we're going to extract out the results. Okay, so if we have a look here, this is our LRT test, our results, lots of significant changes. And so I want to filter the genes out that are significant with an adjusted p-value of 0.01, and then do a heat map. So I need to do assay to get the RLog matrix. And then I need to filter my LRT test results to everything that is 0.01 for adjusted p-value and less, and also is not an NA. Okay, we don't want any NA values. And now we can just ask which, if I look at my sig changes, I have a vector of gene IDs that are significant. So I can just then ask of my RLog matrix, which row names from this, which are the gene IDs, so the gene IDs are the row names, which of these are in my significant changes, and then I'm going to subset based on the result of that to get my sigmat. Head, sigmat. Okay, and so I'm just looking at the first 6 results, and you can see I just have the RLog scores for all these significant values. I'm then going to create my, if I'm going to do a heat map, I want to do my annotated data frame. Okay, so if I do look at cold data, right, this is my cold data. It has 2 things. And so we actually do, this is a nice little, another little trick. So if I just want to look at the tissue, I just want to annotate the tissue. I don't care about the size factor for this plot. So if I do that, it returns a vector. Because it's like, well, all you've got left is 1 column. So we're assuming that that 1 column is all you want. And we'll just simplify it down to a vector because simplification is great. But pHeatmap needs a data frame. So I can then add this argument drop equals false, and then it keeps it as a data frame. And so we can then actually, this is a specific kind of data frame, which is a cold data frame, but we want a standard data frame. So we do as.dataframe, and that becomes our ANODF. Easy. Okay, and so you can see it's lost some of this extra text because there was a specialist data frame. Now it's just a regular boring R1, which is what pHeatmap likes. And we can then draw our pHeatmap. We just got a question from Charles. What would happen if you didn't filter out p adjusted as NA when we try that? So I'm going to create this, and let's remove that. So we're just filtering on less than 0.01. So if I did the subsetting, I'm going to call this sigma2 then. This is sigma2. And then we can draw it again, but we'll do sigma2. To be honest, I didn't expect that to work. I was expecting it to give me an error. So okay. So that's why. So yeah, so we have a good couple questions there from Charles. And so what would happen? Well, if you have an NA score or an NA value, quite often either these counts are super low or they're 0, or they might be NA values in themselves. Then if they get passed on to the P-Heatmap, if it's doing any kind of Z-score or it's doing clustering, if there's any NA values, it will break. In this data set, actually, though, I think I must have already filtered out the NAs previous to putting it on GitHub. So yeah, there isn't any NA. So when I asked sum is .na, well, this is like, this is a bunch of true falses. If you do sum, it counts how many are true. So there's no NAs. Do you filter out the NA because you have multiple comparisons? What happens if it's an NA in one of them and in the other two, there's actually a meaningful difference? That is a good point. There is a chance that you throw out some data that is meaningful. What I would say to that is you will pick that up in pairwise comparisons. So this kind of genome-wide view where you're comparing across multiple groups, you are unlikely to be doing just that. Like the clustering and stuff like that, you're unlikely to be doing just that. You'd also probably be doing some pairwise analyses as well. And so if something pops out the pairwise analysis that's super significant, you can still follow that, right? And those genes will still be there, even if they're NA in another comparison. So that is kind of the answer to that. With these kind of global things, the filtering and trying to hone down, it's just never going to be perfect. And so sometimes you do penalize against potentially interesting things, but on the whole, it's typically worth it. And it's typically not worth parsing things like at such a fine level to make sure you can eke out every single potential comparison. I hope it makes sense, Charles. Okay, so next we do PCA and we want to get loadings. So we want to extract the influence of PC2 and produce a heat map as above, but now ranked by your PC2 rotation / loadings. Okay, so to do that, we need to do PR comp. Okay, and so if you remember, we do the transpose of the RLog matrix before it goes into PR comp. And then we're going to do PC2, so we get rotation 2, rotation, column 2, and in this case, we're just going to do decreasing. It doesn't matter which order you do it, but what we're doing here is we're doing sorting so we can get a rank. Okay, so this is a nice vector with the PCA scores, and then the names of the vector are the gene names. So that means we can use the names, we can match them to the row names, and then use that matching to reorder the significance matrix. Okay. So this reorders using match. We can reorder the significance matrix by saying, hey, we have this vector of names we want to know, and it's ordered in a specific way. We want our matrix to represent that order. So we match the row names against this vector and then reorder based on that matching. Okay, and so then when we do that, we create a new matrix, which we can then put the heat map of. And if we remember, PC2 is really all about kidney, right? And you can see that trend is really present here. Okay, and so you have at this end of the PC, you have the genes that are very much downregulated in kidney. And at this end, you have the genes that are very much upregulated in kidney. Cool. So this next step is getting into something that we haven't really directly covered that much, but we've kind of hinted at a lot of the pieces around it. And so I asked, produce a PCA plot of the Go terms. So we're going to subset to a specific set of genes, and we want to do the heart development genes. And so yesterday, I mentioned that you can get Go terms from the org-mmegdb object, okay? And so you have to extract the genes from this and then subset based on that. So we can load in this org-mmegdb object. Okay, and so this is a database with everything about mouse. And we're going to use the select function. And so to break this down, we're saying I want to select within this database. I have a key type, which is a Go term. Okay, so what they call Go terms in this database is Go all. This is what this Go term is called. This is the key. And I want to get back entree IDs. Okay, so I want to get back gene IDs from this. So if it's just working now. So if I now look at head heart development, I get back Go terms, right? Where the evidence for it was, what kind of ontology it is, and the entree ID for all of these. And so with these IDs, I can now subset our I'm just going to first I'll do unique just in case there's any duplicates. And then here, what we're doing in this operation up here is we're saying which ones from the RLog tissue are contained within this list of genes. Okay. And I want to subset to those ones, and I want to do a PCA. And so now, it's actually slightly different between my versions here. It's interesting. The majority of the variation is in PC1, and you can see that this Go term is associated with what would be with kidney, essentially. No, it's associated with heart. It's specific to heart. Okay, and so if we remember back to our overall PCA, let's see if I have it here. This one. Right, nothing corresponds to, I guess PC2 corresponds to just heart. Yeah. I'm not sure if these are labeled correctly. I think PC2 is supposed to be kidney. Okay, anyway. Yeah, it corresponds to just heart. So we're able to start to pass down and do some interesting things with specific groups if we want. You can see how using the skills we've learned, we can actually start to really break things down and ask very specific questions. Yeah, and so then, we have a little bit more to go. We can use, The next question is to do a heat map of this Go term. Okay, so we do something very similar here. We do select again. In this case, we're going to do 3 different Go terms. Okay, associated with either heart development, liver development, or kidney development. Okay, and so this is just kind of repeating the same thing over and over again, but we're essentially selecting genes based on specific Go terms. Okay. Then what we need to do is create an annotated object, a data frame to annotate the rows of our heat map. And so within this data frame, we're saying that the heart dev genes are equal to the ones that we found in the heart development Go term. And so if I have a look at this, what this looks like. There we go. So we have this big list of genes. And you can see this one is a heart dev gene, but it's also a kidney dev gene. It's in both of their Go terms. And so this is an alternative way to add the annotation with a true false list. We can also add custom colors. Okay, so colors correspond. If it's false, it will be white. If it's true, if it's part of the group, it will be a specific color. Okay. And then lastly, we can draw the heat map. Okay, and so I'm going to zoom in a little bit on this just so you can see what the point of all this was. And so you can, using stuff like this, you can then start to see how these different specific genes kind of group together and how specific gene sets correspond to different clusters. Okay, and so like we have this kidney specific group up here. And this corresponds to previously known kidney development genes. So this is a nice way to validate your data, right? We have the Go terms that we think are kidney development, and it corresponds in this large group which are behaving in a kidney specific way. Interestingly, things like heart development there's a lot of overlap in the heart development specific groups, but there's also some with kidney. And then liver isn't actually near the liver. So that could be something to look into further. Maybe something's funky with this liver data set. Maybe we need to pick a different gene set. Okay. So next is just clustering, and we're saying just do the same, but we want to look at the 3 clusters. So this is similar to what Charles was talking about earlier, is what happens if we just did the 3. Okay, it will look very much the same as it will be similar to when we've optimized the clusters. Okay, and you can see how the generalized cluster-wide view is specific like this. You can break down and then ask if you can draw a heat map ordered by cluster. And so you can break this object down, this K, see what's in K. There's lots of different options. There's things about the tree row that we care about. K means. So this contains the cluster information. Okay. And so we'll turn that cluster information into a data frame. It looks like this. And then we'll reorganize by that data frame. Okay. Okay, that is that. So we're going to head on to the final section. Yeah. So that's a good point. I feel the most useful workflow would be to perform clustering with a K means of choice and list the top marker genes in each cluster to check whether the clustering makes sense. Yes. Yeah, that would be a good way to do it for this. We could then see if these liver-specific groups are falling into these specific groups here, see if there's overlap. But it all depends on what your data set is, because there might not be something that is a nice standard to compare to. You can see how even with this is a nice data set that we use a lot for like training, and it doesn't correspond necessarily to liver development. Maybe that's because at this point the liver's already developed. I don't know enough about this data set to know exactly why those overlaps might not exist. But that's when the experts with their specific data can ask the better questions about how to analyze, what gene sets to use, and things like that. And so, speaking of gene sets, we are going to switch things up